Assalamu alaikum guys. So today I'm here with a new video, Lepidium part 2. As in, if last part I have already explained you that what is Lepidium. I've already given you an overview about Lepidium, which is also called Maca. Well, today we are going to discuss about its nutrients. What are the nutrients within Lepidium, how they are extracted from it, and is there any side effect of Lepidium on a human body. So we'll discuss them one by one, and let's see the content first. So first of all, I have nutrients within Lepidium. Second is extraction method. Well, after the extraction method, we have its nutrients, which are glucosinous, carbohydrates, Meyenii, Meyenins, thiodentynes, alkaloids, plant proteins, lipids, and also at the end effect of Lepidium, well, which is also known as side effect of Lepidium. So let's start our presentation with what are the nutrients, or let's talk about the nutrients within Lepidium. So Lepidium consists of many helpful nutrients which are quite essential and useful for mankind. And these nutrients do not only imply on human, they are also useful for other living organisms such as animals, of wildly animals. We will discuss them one by one with its benefit and uses. Every nutrient in Lepidium has its own characteristics, so let's start the one by one. Well, they are quite useful with the help of biotechnology nowadays. Lepidium is being used for making quite those medicines which have a good effect on cures and cancers and any other stuffs. So let's talk about the extraction method. So water soluble polysaccharides were separated from Maca aqueous saccharide, which is also called MAE. The crude polysaccharide were deproteinized by swag method during the preparation process of Maca. Polysaccharides, amylase, glucomylase effectively removed starch in polysaccharides. Full PDM Meyenii polysaccharides LMPs were obtained by changing the concentration of ethanol in the process of polysaccharides precipitation. All of the LMPs were composed of ramoles, arabinose, glucose, and lactose. Antioxidant activity tests revealed that LMP60 shows good capability of scavenging hydroxyl free radical and superoxide radical at 2 mg or milliliter. The scavenging rate was 52.9% and 85% respectively. Therefore, the results showed that the Maca polysaccharides had high antioxidants activity and could be explored as a source of bioactive compounds. So this was the extraction method that I've already told you. Now let's see that this is a picture of Lepidium in powdered form. Well, it's not extracted in this, but the powdered form is essential to eliminate the nutrients from it. So the first, and let's go for the glucosinous. So this is one of the nutrients that we extract from Lepidium. So unique secondary metabolites, glucosinolates, S-glycosanoprolose, thiohydroxymates are naturally occurring S-linked glucosides found mainly in Brassica plants, which are essentially Lepidium. They are enzymatically hydrolyzed to produce sulfate ions, D-glucose, and characteristic degradation products such as isothiocyanates. The function of glucosinates in plants remain unclear, but isothiocyanates possessing a pungent or irritating taste and odor might be associated with plant defense with microbes. Isothiocyanates have been studied extensively in experiment in vitro and in vivo carcinogens model for the cancer as an chemopreventive properties. So glycosinolates is usually used in order to make cures for carcinogens and in chemotherapists in such as ways. So the beneficial isothiocyanates, glucosinolates that are functional for supporting human health have received attention from many scientists studying plants breeding, plant psychologists, and plant genetics and food functionality. This review presents a summary of recent topics related with glucosinolates in Brassica family, along with summary of the chemical metabolism and gene of glucosinolates. In brassicas, the bioavailabilities of isothiocyanates from certain functional and glycosinolates and the importance of breeding will be described within emphasis of glycosinolates. So in these pictures we can see that glycosinolates can be used in yeast, E. coli, which is usually bacteria used for producing yogurts and other things, and brassicas plants. In the production of brassica plants, glucosinolates are essential, and also in the production of tobaccos, as we have seen in this slide, that it is also used in carcinogens models. So the second nutrients in Lepidium we have is carbohydrates. So carbohydrate is really essential part of your body. It gives strength to your brain, your digestive systems, and other stuff that are really important for you. So carbohydrates are essential hydrates of carbon. They are composed of carbon and water and have composition of CH2O. And carbohydrates, polyhydroxyaldehyde, ketones, and their derivatives. Carbohydrates have the following basic composition. Carbohydrates have spare protein, so that protein can concentrate on building, repairing, and maintaining body tissues instead of being used as energy source. So for fat to be metabolized properly, carbohydrate must be present. If there are not enough carbohydrates, then large amounts of fat are used for energy. Carbohydrates is necessary for regulation of nerve tissues and is the only source of energy for the brain. Certain types of carbohydrates encourage of growth of healthy bacteria intensity for digestion. Some carbohydrates are high in fibers, which help prevent constipation and lower the risk of certain diseases such as cancer, heart disease, and diabetes. Functions of CHO. Now basically we'll discuss that we'll discuss macamides and macaines. Before that, let's see the structure of carbohydrates. Well, here is the structure of carbohydrates. What is basically a carbohydrate? So as we can see, it contains of one carbon chain with CHCO2, which is attached to many of H, which is again linked to OH and water or compounds. I guess it has 1, 2, 3, 4, about 4 to 5 water molecules attached to it. So let's talk about macamides and macaines. So macamides are characteristically found in Maca, Lepidium, Meyenii, Valper. Fatty acid derivatives are also an important type of constituent in Maca, since they not only relate to the biosynthesis of macamides in the post-therapist process, but also possess some bioactivity to study their comprehensive profiles in Maca tubers possessed via the air drying method. Ultra performance liquid chemotherapy, quadruple time to flight of mass spectrometry, UPLC, QTOF, MS analysis were performed to identify macamides fatty acid molecules. Their contents in Maca tubers that were processed in via air drying and freezing drying respectively were further quantities using high performance liquid chemotherapy analysis comparing with 8 macamides and 3 fatty acids reference standards. So a total 19 macamides, including 4 normal ones and 16 fatty acid derivatives found in Maca for the first time were identified. Quantification analysis results showed the 8 macamides with content ranging from 31.39 to 1163.9 microgram over gram on dry tuber, and fatty acids from 18.17 to 181.99 microgram in the air dried Maca. But there were only 3 macamides and only fatty acid detected were with very low contents, about 3.97 to I guess 3.436 microgram in the freeze dried Maca. The results demonstrated that the air drying method can increase the accumulation of macamides and fatty acids in the metabolism of Maca in the post-therapist process. The biosynthesis of 2 types of macamides, for example, N-benzyl oxo-octadynamide and N-benzyl oxo-octadecatrinamides was further elucidated in detail. These results provide more valuable insights into the phytochemicals of Maca, which is helpful to explain its health benefits. So these are the structures of Mecamides and Mecameyenes, which is also an enzyme, which we know that is basically an enzyme which is essential for metabolism and can be increased through dry aging in Lepidium. So let's talk about thiodentoins. It is also a nutrient that is found in Lepidium. So reactive oxygen species ROS causes oxidative damage to cellular membranes and macromolecules, leading to disease such as cancer. Antioxidants are chemical substances that scavenge free radical and ROS and can be used as an alternative therapeutic approach to treat diseases. In the work, we evaluate the in vitro antioxidant activity of series of thiodentoins, which five derivatives shows promising radical scavenging ability against stable DPPH. The IC50 values range between 100 to 270 micrometer, and the best antioxidant potential was observed to thiohydrantones containing amidazole group in the side chain. Kinetic analysis of antioxidant activity indicated fast, intermediately, and slow behavior, while quantum calculations showed that the lowest IC50 is related to the best dominating electron ability of the molecule. The molecule docking study for the best derivatives against the five relevant targets involved the redox, hemostasis, LOX, MPO, NOx, and XOX, suggested that the thiohydrantoins rings act as a pharmaphoric group in the interaction with catalyst residue in the active sites, demonstrating the thiohydrantoins containing amidazole group in the side chain can be promising compound as an antioxidant, which is usually used in cancer treatment, which is also essential for other diseases that are yet to be discovered, that cures are yet to be discovered. So this is a structure of thiohydrantoin. As we can see, a benzene ring consists of two NH and an oxygen, and which is also attached to a sulfur. So the next nutrient that we have in our list is alkaloid. The term alkaloid, means alky and oid means like, was proposed by W. Mincerin in 1819 for basic nitrogen containing compound of plant origin. It may be defined as a physiologically active basic compound of plant in which at least one nitrogen atom forms a particle of cyclic system. So in this slide we can see that the structure of alkaloid, that we have a lot of benzene rings attached to each other, but they have different nitrogen groups attached to it. And forms of part of water cyclic nitrogen atoms forming particle cyclic system, in which we can see this all of them. So alkaloids are cyclic organic compounds containing nitrogen in negative state of oxidation with limited distribution among living organisms. Form of alkaloids. Well, there are several forms of alkaloids. So these are free bases, salts with organic acids, for example oxalic acid, acetic acids, salts with inorganic acids, for example HCl and H2SO4, salts with special acids, for example meconic acid in opium, cunic acid and quinquina, and glycolial form, for example solanine in solanum. So the next part is plant protein. In addition to providing a building block for healthy muscles and tissues, plant protein may be beneficial for weight loss. Plant-based proteins tend to be lower in calories and fat than animal protein, but high in fibers and essential nutrients. Here we can see the structures of plant protein. So here we can see there are two amino acids attached to each other in front of each other like this, and they are naturally attached to each other which contains a high peptide bond which cannot be break, and it also contains huge quantity of water within it. You have to make it more moisture and give it more beneficial and accuracy in the human body. So the benefit of plant-based proteins includes increased intake of fibers, low risk of cardiovascular disease, and some types of cancers, and benefits of weight management. Additionally, plant-based proteins do not contain some of less healthy compounds found in meat, including saturated fat and cholesterol. And basically, as we can see, they are huge. They have a high quantity of fibers and low fat of fat in them, so they are very basically important for a person who usually hits the gym and do his workout and everything stuff, so he should use these plant proteins. So lipids. In lipids, the synthesis of fatty acid take place in the chloroplast, and the fatty acid synthesized is prokaryotic type. In plants, the structure of membrane lipid is different from the eukaryotic cells. The membrane of chloroplast are essential, formed by galactolipids. Lipids of seeds are essential composed of DAG. It would be interesting to describe their system. Their synthesis during the maturation of the seed. Some plants contain in their reserve lipid unconventional fatty acids such as gamma linoleic acid and borage-officinalis L, short chain fatty acid C12 and C10 fatty acid with very long chains, and fatty acid that are cyclical. All of these fatty acids can be industrial or pharmaceutical application. Here is the structure of that plant lipid. This is, as you can see. So our final slide is that what defect of Lepidium or side effect of Lepidium. So there is no such nothing that negative side effect of consuming too much Maca. One retailer says a small percentage of customers having reported like issue jitter feelings, insomnia, and mild digestive issues, typically for the first time users, those who adjust to the product. But in last presentation I've already told you about that the pregnant or the women who are having and babies should avoid this, because of this there are some properties that could awaken mature hormones which are not good for the baby, so they should avoid this. But for a normal person there is no side effect for using Lepidium. Actually, as we have read its property, it is quite useful for sexual drive, for exercise, for daily problems. For it's also used for major problems such as cancers, digestive problems. It has a lot of good things, so we should use it. So this was my presentation, so hope you guys liked it, and don't forget to subscribe and share the channel. So see you guys, and Allah Hafiz. Thank you.